Сегодня мы проводим с вами семинар по вопросам, связанным с демографией, наверное, так можно сказать, а может быть более узко по вопросам активного долголетия человека. На медицинском факультете эта проблема очень важна. И сегодня у нас в гостях ведущие специалисты, работающие в России в этой области. Мы прослушаем именно в России работает прекрасный специалист с высокой репутацией международной, профессор Франчески, Клаудио Франчески. Он любезно согласился прочитать лекцию. А кроме того, здесь присутствуют ученые, работающие в Москве, в Нижнем Новгороде, в других местах нашей страны. Я боюсь назвать, не знаю, вы по-прежнему там же, да, в Сыктывкаре, нет? Вы уже в Москве. Вот поэтому я и споткнулся. Когда-то вы были там, думаю, неужели все еще там? Да. Ну и многие сотрудники МГУ, которые интересуются этим вопросом. Я два слова хочу сказать дополнительно. Передам сейчас слово организатору этого мероприятия Константину Юрьевичу Кулебякину, доценту нашего факультета, который организовывал семинар, и он будет его вести. Я же хочу сказать, что проблемы долголетия для меня выглядят таким образом. Ну, известно, что продолжительность жизни зависит от многих факторов, но такая средняя продолжительность жизни мужчин, женщин в каждой стране очень хорошо коррелирует с ВВП. Вот внешним этим продуктом, с успехами в экономике, параллельно и медицине, а параллельно и в условиях жизни, в экологии и так далее. Вот это то, что так вот мне представляется. Но есть еще проблема индивидуальной продолжительности жизни, которая зависит уже от каких-то факторов, которые едва ли мы можем все перечислить или которые мы понимаем. Мой друг и учитель, он читал мне лекции, мы с одной кафедры, Владимир Петрович Скулачев, верил в то, что есть ген, который определяет окончание жизни, ген смерти, условно названный. И у каждого вида есть своя продолжительность жизни индивидуумов. У людей это около 100 лет, а у каких-то животных 300, у каких-то 1 день, 1 неделя, 1 месяц. И он искал этот ген. И он ушел от нас 2 года назад. Я с ним постоянно общался на эту тему. И в конце своих исследований он сказал: ну вот мы не нашли такой ген, и никто не нашел такой ген, но пройти этот путь надо было. Я не жалею, что я проводил эти исследования, потому что все в науке надо проверить. Да, похоже, что такого гена нет. Это сказал Владимир Петрович года 3 назад в разговоре мне. И он написал это в очень хорошем журнале обзор физиологических наук. И вот при этом, конечно, тема не закрыта. Как всегда в науке, всегда еще есть что проверять. Но есть и другое высказывание очень мудрого исследователя еще в 90-х годах, которое мне ближе вот персонально. Он сказал, что жизнь начинается с регенерации и завершается, когда прекращается регенерация. Невозможна жизнь без регенерации, но и смерть неизбежна для продолжения жизни. Между полюсами этими начало регенерации и завершением регенерации и находится продолжительность жизни. Вот мы эту гипотезу проверяем. И вот я хочу сказать, что регенеративная медицина, она наука молодая, многие ее могут не знать. В общем-то, она в этом веке возникла как наука. Но в этом году появился национальный проект в нашей стране. Благодаря усилиям ученых всех нас, в котором в одной строчке написано «регенеративная медицина» и активное продление активного долголетия человека, активного долголетия. Это уже та тема, которую мы сегодня будем обсуждать, мне кажется, больше. И это вот вместе, они финансируются, эти две науки вместе. И финансирование довольно серьезное заявлено. Даже боюсь произносить эту цифру, вы можете в нацпроектах это увидеть. Но очень радостно, что наше руководство страны обратило внимание на активное долголетие, продление активного долголетия человека и ожидает какие-то научные проекты, которые будут подсказывать, как продлить это долголетие. Ну и все это объединено с регенеративной медициной. Ну, даст Бог, и мы будем полезны, те, кто занимается вопросами обновления клеток. Клетки обновляются всю жизнь. Тонны клеток производит человек за свою жизнь. Десятки тонн клеток даже. И они погибают, и они образуются, и когда прекращается обновление в каком-то органе по какому-то механизму, который мы хотим понять, тогда и страдает этот орган и оканчивается жизнь. Вот такая точка зрения у нас, поэтому мы очень близки все друг к другу, тем, кто занимается долголетием, и те, кто занимается регенерацией. Я очень признателен Константину Юрьевичу, который взял на себя труд организовать этот семинар. Благодарю всех, кто пришел на эту беседу. Она очень своевременна. Впервые в нашей стране финансирование получено под эти науки. И, может быть, кто-то из вас, заинтересовавшись этим, более активно вовлечется в эти исследования. Спасибо большое нашим гостям, приехавшим из других городов на этот семинар. И особая благодарность профессору Франческо, который свою науку развивал в Италии, потом получил мегагрант и сейчас, как я знаю, в Нижнем Новгороде занимается этой работой. Прошу вас, Константин Юрьевич, это ваш семинар, вы его ведете, а я поблагодарю всех, пожелаю всем доброго здоровья и активного долголетия.
Всеволод Арсеньевич, большое спасибо за вступительное слово, и я вообще бы хотел бы высказать огромную благодарность Всеволоду Арсеньевичу и всему факультету фундаментальной медицины и нашему всей университетской клинике за помощь и в организации, и, там, разумеется, без наших коллективов этого было бы невозможно, но и огромную благодарность я бы хотел высказать и профессору Клаудио Франчески, который согласился прочитать такую лекцию, и Алексею Москалеву, который помог это организовать и вот наладить научный контакт. Поэтому я всем высказал благодарности. И теперь представлю нашего уважаемого лектора, профессор Клаудио Франчески, почетный профессор университета Болоньи, профессор Государственного Нижегородского университета имени Лобачевского и является одним из ведущих специалистов в области биологии старения, является автором концепции Inflammaging, о неразрывной связи иммунной системы, воспаления, процесса накопления старческих нарушений и вообще очень продуктивный ученый с широчайшим кругозором и хотел бы еще раз поблагодарить его за то, что он согласился, и предоставить ему трибуну.
Thanks a lot for this invitation, and to the dean, the faculty, the students that are present here today in this very important and famous university. Thanks to Alexei Moskhalov. So I come from Bologna, Italy. We are one of the leading countries regarding the aging, because the average life expectancy for women is about 84, 85 years, and for men we are always a little bit less. I'm so sorry, but is 81 or 82. Here the life expectancy in Russia is a bit lower, but I think that my mega grant and the new project that were mentioned will help in improving this data. And I can start. Okay, so no, this.
Okay.
I cannot start.
No, you can. Right, please.
Okay, so let me remind you that I am the editor-in-chief of number 2 journal in the world regarding aging. The first is Nature Aging. The second is my journal, Aging Research Reviews. The impact factor is quite interesting. And Alexei Moskhalov is part of our team as an associate editor. And the rejection rate is quite high, but of course, this is a rule of the game. I would like to suggest that my lecture today in this beautiful hall is a sort of legacy to Ilya Mechnikov, because this great guy was studying macrophages, inflammation, gut microbiota, and centenarians, exactly some of the topics that I will address today. So it is a sort of ideal continuation of this great guy. This is just to illustrate the mega grant that we got in 2018, and of course here these are the people and the topic that we should address: chronological versus biological clock, Parkinson disease, Down syndrome, Inflammaging, and healthy aging markers. So, and now we will address, I think, in the future, rejuvenation strategies that were mentioned. And well, what is interesting is that in this mega grant there was a great combination, because I did not understand anything about artificial intelligence, because I am basically an immunologist. But in Nizhny Novgorod, the people were very good in artificial intelligence, machine learning, and deep learning. So it was a great combination, I think, because artificial intelligence should be applied. It's a tremendous opportunity to apply AI to aging and longevity. And this is exactly what we are trying to do. Aging longevity and age-related diseases are quite complex. I am sorry for the students, but they are quite complex. Historically, and they are dynamic phenomena changing according to time and space. So aging in Russia can be a bit different from the aging in Bologna. And Homo sapiens age at different rates at the population, gender, and individual level. So you can understand how big is this complexity. So just to start, to give you an idea, I would like to remember this paper that I published with some great friends like Tom Kirkwood, Caleb Finch, Stig Olmos, and Rudy Westendorp. We addressed the role of non-linear stochasticity and non-linearity in the aging process. So the idea is that, as illustrated in this figure, that individual mortality of worms, in this case Cenorhabditis elegans, a great model for studying aging, of the same lab, same genetics, given the same food, kept at the same temperature in the same room, as you can see, change enormously. And if you change something in order to let living longer, the same curve, you obtain the same curve. So this means that stochasticity is an integral part of this phenomenon. And this is illustrated here, in this case, not in worms, but in mice. This is a typical curve of mortality in mice. So some mice die quite early and other much later. And if you put them at, say, if you let them eat less, the lifespan can be increased, but the curve remains the same. And in this case, so we published this paper together with the people in Nizhny Novgorod regarding trying to disentangling the part of aging which is deterministic, stochastic, and non-linear. So we assessed the signal-to-noise ratio. We found that about 90% of the age is age-dependent. DNA methylation variability is deterministic, but 10% is stochastic. And the methylation level itself is more than 50% of the cases vary non-linearly with age. So stochasticity and non-linearity of the process. So the major characteristics of aging are increasing heterogeneity, individual variability, and there is an urgent need to go beyond chronological age. So just to give you the flavor of heterogeneity, this is a paper that I published some years ago on a lot of people, age range from 20 to 113, including 144 centenarians and offspring, and 70 people over 105 semi-supercentenarians, and 308 centenarians offspring. And look, this is the variability of the thyroid function. The thyroid function varies a lot, and look how much it is variable in centenarians, for example. So there is a spreading of variability. And this is another paper where we study the so-called mitokines. So the mitochondrial dysfunction elicits the production of stress response molecules called mitokines. This is a paper. And look, this is a GDF15, a major mitokines involved in the aging process. And you can see very clearly how the variability increases tremendously with age. So we definitely need biomarkers, chronological age. Some people can have the same biological age as chronological age, but there can be people that are younger or older than their chronological age. And this is some of the idea of the clock that we are studying in Nizhny Novgorod. You can see the same concept, green younger or red older. And of course, we can quantify the acceleration of the deceleration of the aging process with respect to chronological age. How to measure biological age in humans? Well, let me remind one of the first papers that I published on this point. Steve Horvath of University of Los Angeles in the US identified the clock. So studying about 500,000 of methylated cytosine in the genome, he identified 353 methylated cytosine that constitute a clock, a methylation clock, which is quite reliable because the correlation between DNA methylation age and chronological age is very high, close to 1, and the error is only about 3 years. And together with Steve, we studied our centenarians and supercentenarians in this paper. And we studied 82 people over 105, so it's a remarkable age even for Italy. And the offspring, 63 offspring, and control of the offspring. And just to make a long story short, the data suggested that semi-supercentenarians, so people over 105, are about 8.7 years biologically younger than their chronological age. But also the offspring are younger, at about 5 years. In the control of the offspring's chronological age and biological age coincides. Just to give you an idea of the sensitivity of this clock, we studied Down syndrome. Down syndrome, we knew before that Down syndrome is a syndrome where there is some sign of accelerated aging. But in this paper, together with Steve Horvath, again, we found that on average, these people are about 6.6 years older than their chronological age, but looking at the different organs and systems, the brain is exactly as predicted the organ which is much older, about 11 years older than their chronological age. But we have other clocks, for example, the N-glycan clock. For example, in the immunoglobulin, there is a part which is made of carbohydrate, and the last part, say the sialic acid and galactose, are lost during aging. So this is a very important clock, and this is a very recent paper that we published on a consistent number, 246 individuals with Down syndrome from 3 European countries, and the biological age of adults with Down syndrome in this case was on average about 18 years older than chronological controls, non-Down. But one of the most interesting outcome of this paper is that this accelerated aging is detectable from early childhood. So this suggests that some clocks start before birth, but in this case, Down syndrome age within the uterus, when they are within the uterus. So when they are born, they are older than other newborns that are not Down, and the rate of aging in these people is the same, but they start older since the beginning of their life. This is a work, we established a new clock. I'm sorry that the slides is not so clear, but we in this paper we identified a new clock, and the genes are in blue there. And again, with this new clock, using methylated cytosine of the genes that are in blue, we found again that the biological age is significantly higher in 62 Down syndrome subject and lower, as expected, in 106 centenarians and centenarians option. So two clocks gave practically the same results. These are now some result of what we did in Nizhny Novgorod. You can check these and go, and so you can have an idea of what the clock is. You have time to okay. So this is one of the papers that we published a couple of years ago. We constructed a machine learning based cognitive clock that predicts chronological age with a mean absolute error of 8.6 years, which is not so bad. So we employed 3 cognitive tests: differentiation of shades, campimetry, arithmetic correctness, and detection of reversal lectures. And we demonstrated the presence of correlation between this cognitive clock, phenotypic, and epigenetic age acceleration. So they are correlated altogether. And you can see here that you can see here, for example, that the best correlation is with these clocks, and here is the machine learning, the result of the test. These are the tests here, 1, 2, and 3, and the data suggests that the results are non-linear. You can see here that they are non-linear, and that there are a lot of difference between individuals. These are performing very well, and these, of course, at the bottom perform very bad to the different tests. Okay, so this is the novelty, I think, that we did an improvement, and using new cognitive tests, in particular Stroop test, and another test to check the recent memory. So 5 tests, 5 cognitive tests, and this is the results. You can see how the correlation with age, and using explainable artificial intelligence SHAP values. So SHAP values tell you how much each feature contributes to the model prediction. And you can also check the local explainability by the so-called waterfall plots. If they are in red, the prediction suggests that the molecule that you checked is higher, in blue is lower. And these are the results in men and women, and the importance of the different clocks. But what I would like to show with more attention are these waterfall plots. And you can see that, for example, in this case, there is at the top, this female was 47, and the cognitive age was 65, so in accelerated aging from a cognitive point of view. And these are the molecules that and their importance. And here the test, sorry. Here you have a female of 47, cognitive age 37, so 10 years less. And these are the tests, the importance of the test, how the test impact on this cognitive age. And so I think that this new clock is very powerful, and I think that we are testing this in how many, Misha? 1,000? No, 10,000. 2,000. So a lot. And next year, if you like, we will present here the results. This is an interesting paper that I got an European project on Parkinson, 6 million euros to check the importance of aging and inflammation in Parkinson. And this is one of the papers that has just come out in June this year. And you can see that the title, Plasma Proteomic Identify Biomarker Predicting Parkinson Disease up to 7 years before the symptom onset. And these are the eight molecules. The 8 molecules: granulin, mannan binding lectin, this one, prostaglandin H2, complement 3, and so on. And what is interesting is that these molecules are largely involved in neuroinflammation, because we know that how important is neuroinflammation for Parkinson. And this gives you an idea of the accuracy of this clock, because, for example, in isolated REM sleep behavior disorders, these are people that have a lot of problem in sleep. They move, they make a lot of movement and so on. But after a few years, they develop either Alzheimer or dementia. And you can see very well that, you can see very well that these are the control. These are the de novo Parkinson that have not been treated at all. So at the starting of the disease, and this is the IRBD, so the people that in few years develop Parkinson. And we can distinguish de novo PD 7 years. We can have 7 years before the onset of the symptoms. And the epigenetic clock, one epigenetic clock that we did in these people suggests that the patient with these isolated REM sleep behavior disorders are older than their chronological age. We studied 28 people of this type, 57 control, negative, and 31 positive control. And IRBD presented a mean disease duration of about 9 years, were followed for about 3 years and a half. And 8 patients, 28%, converted into an overt alpha-synucleinopathy, which is exactly typical of Parkinson. And you can see that the people can be clearly distinguished from the control. Now I would like to present a few data that have been recently obtained in Nizhny Novgorod regarding diabetes, type 2 diabetes mellitus. The database come from the Italian National Research Center of Aging in Ancona, Italy. I was the director of this national center some years ago for 10 years, and I started to study people with diabetes, and 16, 17 practically years later, these are the data that we collected. So we studied about 568 patients, followed for 17 years, and these are the basic characteristics regarding comorbidity, complications, drug, and so on. And this is the workflow of what has been done. So we started with these patients, and there are data processing, and then there is the final data set. Then you can select the best model, and so on. I will not go into too many details because this is the part that the people do. And this is just the final result. So we studied about 100 parameters. And after this complex type of analysis, 6 parameters came out as the most powerful to predict mortality in these people. And the parameters are the following: of course, chronological age, but the other one is brain natriuretic peptide, creatinine, estimated glomerular filtration rate, urea, and CC8. And these are the top here of all the markers that have been analyzed. And you can see that with this marker, you can predict a very different mortality according to the presence or absence. So this clock is very powerful. And we can go to the individual level with explainable artificial intelligence. And you can see here that this suggests that these are the parameters. And from this side means that this increases the probability of mortality. So, okay. And at the individual level, it is very interesting because, for example, it tells you again the importance of each of the parameters, blue and red. This is, for example, a female age 73, disease duration 24, and this woman died. This is the same age, but look how different is the combination and the importance of the different markers. So you can go at a personalized level. This is a guy that is an exception because everything was practically wrong, and he is still alive despite the fact that most of the parameters are red, and is still alive after 16 years. Probably there is another number of factors that may influence survival that are unknown to the model. So there is a space for improvement. So the take-home message is that an explainable machine learning model was developed to predict the mortality risk of patients with type 2 diabetes. The resulting model includes 6 features to be strongly associated with diabetes. The model quality indicators such as the index and the IOC have high values in the test dataset, and you can see how interesting they are. So 0.7, and the IOC is close to 8. Okay, if I have time, I would like to tell something about Inflammaging and the inflammatory clock. I published some years ago, about 24 years ago, I published this paper in an obscure journal, the Annals of the New York Academy of Science. But despite this fact, this paper is now quoted more than 6,000 times. The idea is that with the age we develop a chronic low-grade sterile inflammation. And the hypothesis is that the macrophages are very important, are crucial cells for inflammaging. Some years, while studying the centenarians, we realized that the centenarians have a low level of inflammaging, but also high level of anti-inflammatory molecules. So we suggested in this paper, again published in a not so great journal, but Mechanism of Aging and Development, but today this received more than 2,000 and about 400 citations. So it's not important where you publish, it's important what you publish. This is a message for the young people. So my modest suggestion. So the centenarians are inflamed, but the data suggests that in these people there is an increase of pro-inflammatory molecules. So inflammaging is continuously compensated by anti-inflammatory molecules. And this is a classical adaptive mechanism. Some years later, in 2013, in Bethesda, we launched the so-called geroscience, suggesting that aging is the single most important risk factor for all major age-related diseases, and that we have to address the age-related diseases not one by one, as usual, but all together. So this is the message of this paper. And we identified 7 pillars that are deranged with age and are responsible for aging, but are all correlated with each other. And inflammation is one of them. And together with Judith Campisi, that is no more with us, we published this paper, Chronic Inflammation and its Potential Contribution to Age-Related Diseases. In this other paper that I addressed the problem how if Inflammaging fits one of the most important theory of aging, the antagonistic pleiotropy theory of aging, suggesting that age is not predicted by evolution, and something that can be good when you are young, a strong inflammatory response, can become bad when you are old. So this is the basic idea. And in this paper, I suggested that with age, a derangement of the other, say, of the stem cell regeneration, of epigenetic stress response, eventually converge on inflammation. And in this paper with several friends, David Furman, Luigi Ferrucci, and many others, we propose this Unitarian perspective of the age-related diseases. So you have many possibilities, say chronic infection, physical inactivity, obesity, dysbiosis, diet, wrong diet, and so on. But eventually, the outcome, despite a variety of causes, and the variety of outcomes, at the center is the chronic inflammation. So why? One idea comes from, I am an immunologist, I told you before, and I suggested the idea of immunobiography. So immunobiography suggests that, suppose that this ball here is a person, and according to early life events, the socioeconomic status of the family and so on, according to adults' life events, infections, the nutrients and so on, this ball can go to different valleys, here and there. So this suggests that the enormous heterogeneity that we see in the people, according to the immune response, but many other parameters, depend enormously of all these parameters together. So depends on your biographies. And you can imagine that there is also a liver biography, a brain biography, and so on. Okay, so just to summarize, Inflammaging can be beneficial and adaptive when you are young, and also in centenarians, but can become chronic and detrimental according to the crucial role of your immunobiography, but also, as I said, liver biography, brain biography, and so on. Just to give you the idea of how important is, for example, nutrition, I was the PI of a project with 9 million euros on the role of Mediterranean diet and aging. And eventually we found that 1 year of Mediterranean diet decreased a lot the inflammatory status of people between 65 and 79. And just 1 year of Mediterranean diet reduces inflammaging and has a rejuvenating effect of more than 1 year, according to the clock that I just used. So you can imagine, if you follow Mediterranean diet for all your life, you will become immortal, practically immortal. Okay, so in fact, we studied a lot the importance of the gut microbiota, and we reconstructed the trajectory of the gut microbiota change with age from 20 to 109 years old. This cartoon is a summary. This is a sort of clock because the most common bacterial species like the yellow circle decrease with age, but contemporarily subdominant species increases. So there is with age a continuous remodeling of the gut microbiota. And you know that the gut microbiota send inflammatory signal to the brain, to the kidney, to the liver, and so on. So this is extremely important. So when you talk about diet, you have always to remember that the diet has enormous effect on your gut microbiota. So Inflammaging can be practically read, say, good or bad, according to a lot of variables that I tried to illustrate to you. For this reason, it's important to quantify Inflammaging. And this is the first inflammatory clock that we published 3 years ago, together with people in Stanford and David Furman. And we studied about 50 molecules, and we identified the top 15 that are a very powerful inflammatory index. And these are the molecules that are involved, have been selected. There is a correlation between this inflammatory clock and calendar age, and on the right you can see that there is a very powerful correlation between this inflammatory clock and the number of diseases that the people have. So it works. But the centenarians are much less inflamed than people of about 80 years, the control, and so on. So the centenarians do have inflammation, but much, much less. And in this cartoon, we suggested that in green, so the inflammation is green, is good when you are young, but in the second part of life, when you start the aging process, that has been evolutionary unpredicted because the people were dying before. So evolutionary speaking, aging was not largely predicted. The inflammaging can become older, higher, and can become important for the onset of age-related diseases. Centenarians are in between, are still rather green. Thus, inflammaging is basically dichotomic, but fundamentally adaptive. For this reason, it's particularly important to quantify inflammaging in single person, as we see the difference between centenarians and non-centenarians. And this is exactly the clock that we performed, we set up in Nizhny Novgorod, which is personalized. The study involved a group of 300 people from age between 21 and 101, including end-stage chronic renal disease patients. And we started from 46 molecules, and we selected the top 10 that are showed here, and with explainable method allow to check at this clock at the individual level. So, as expected, the people with chronic renal disease have a much higher inflammatory index, and look here, as I said, with the local explainability, you can check, for example, this guy in the left is age 21, inflammatory age 20, and you can identify the importance of the different combination of inflammatory parameters in this guy. This is a guy of 86, inflammatory age 85, and this is the combination of parameters. But look here, this is a person of 60, inflammatory age 41, so younger, and the peculiar combination of inflammatory markers. The same is for this guy, which is older as inflammatory age regarding in comparison to his or her chronological age. So we can. So now we have personalized Inflammaging, and we have published this paper just published. Explainable artificial intelligence in aging clock models. And what is interesting is that both clocks, the one of Fuhrman and the one that we discovered, works very well. But apart from CXCL9, which is very important in both clocks, the markers included in the two inflammatory clocks are different, suggesting that Inflammaging is not only personalized, but much more complex than we previously thought. So now we are working on a combination of the two markers, and maybe some of you can have another idea and add another markers that can be even more interesting. So I would like to finish talking of the fact that of the multiple organ clocks. So in this study with the Chinese colleagues, we studied 4,000 people between age of 20 and 45, so young people. And in each person, we measured more than 400 parameters, including biochemistry, immune parameters, body composition, electroencephalography, facial skin features, gut microbiome features, and so on. So this is just, so we took blood, stool, physical examination, imaging, and so on. And you can see that there is an individual variability of the aging rate of organs and systems. So here is the kidney, cardiovascular system, liver, sex, skin, immune, metabolism, and so on. So the body is a mosaic of clocks. One possible explanation is the presence of multiple cellular clocks, being organs and systems composed of a mixture of different cell types. But of course, this is in addition to this phenomenon is different in each person. And you can see here this is a particular group of people of about 400 people, and you can see, look at the different parameters here, and the different colors suggest a different intensity of the marker. You can see how variables is the combination of these parameters in 481 individuals. So the biological aging rates of organ and system are diverse. There are multiple clocks within the body, and we, using the data set of this mortality US National Health and Nutritional Examination Survey and the longevity in the Chinese Longitudinal Health Survey, we were able to show, for example, to predict on the basis of this clock that several years later, a young guy that has, for example, the liver which show an accelerated aging, 10 or 20 years later, there is a great possibility that he will develop a disease exactly in this organ. So it's predictive. So organs and systems are aging at different rates. Specific biological age could predict the disease of the corresponding organ, as I just said. Well, of course, there is a different genetic architecture, but I have no time to go into details. Look at this paper. This paper is not mine. This is of a strong group published on December last year. They took blood organ-specific protein from 11 major. They identified blood organ-specific protein from 11 major organs in more than 5,000 adults across the human lifespan in different cohorts. And the 20% of the population show strongly accelerated aging in one organ that confer 20-50% higher mortality risk and risk of organ-specific disease. Exactly what we found. In this case, they found the same using proteomics. And for example, accelerated brain and vascular aging predict Alzheimer. So this is what we have just published, organ-specific biological clock from intra-individual clock, inter-individual, and organ-specific, so what we call ageotypes. And this allows a personalized anti-aging approach. And this is the last paper that I checked in the literature. They took the UK Biobank samples, and they studied more than 50,000 people, 53,000 UK Biobank participants, and they identified organ aging specific signature, and this model predict organ specific disease and risk of death, and reveal that chronic diseases reflect faster aging in specific organs. So this is the message. So our papers 2 years ago, and these other papers that I have showed you quite recently. This is October 2024, so just came out. So this suggests that the idea that there are specific organ-specific clock and diseases is very powerful. I would like to finish with this. Alexei Gorban asked me to write a comment on his paper, Dynamic and Thermodynamic Model of Adaptation. I do not understand anything about thermodynamics, I have to confess, but there was a part of the paper that I could understand, and so the idea is, look, these are what I wrote about this paper. I greatly appreciated one of the conclusions of Gorban paper, when application of the adaptation theory. So adaptation is a crucial word of whatever I told you. Developed in the paper are suggested, and is explicitly said that successful aging is not connected with excessive capacity to cope with the stressor, but rather those with the capability to afford intermediate response to stress. It is neither too weak, nor too strong in relation to the environment. And the second, as I noticed several times, a basic work of the Gorban paper is adaptation, an extension of the rigorous refinement of ancestral adaptability in biological terms, which is assumed as the key to merge biology and physics, thermodynamics, and for the development of the new discipline of adaptation engineering. I think that it is not by chance that adaptation is also the basic conceptual background of Inflammaging, as I try to summarize in this paper, but also for you today. Thanks a lot for your attention.
Thank you very much for your presentation. Question about I understand that you are studying for some markers of inflammaging. Have you found some correlations of biomarkers of cancer and biomarkers of inflammaging or brain age correlation with cancer? Because cancer is sometimes connected with inflammation, and inflammation has found such correlations.
So thanks for this question. I am sorry that I did any study on this point, but a friend of mine, a very famous scientist, Alberto Mantovani, he suggested many years ago in a famous paper in Lancet that maybe you know better than me, that inflammation is a very important component of cancer. So usually, thanks for this question, because usually people in the cancer research, as far as I know, try to kill the cancer cells. So the target is to kill cancer cell. Okay. And there are a lot of possibilities that are, but they do not take into account that the environment where the cancer cell grow and differentiate in order to metastasize, for example, is an inflammatory environment, as you said. So now there are some papers coming, recent papers, suggesting that probably the best way to treat a cancer is not only use the most new and powerful technology to kill cancer cell, but also to try to change the environment, to reduce the inflammatory environment, which is the environment which let the cancer cell to grow. So the answer to your question is, thanks, because I think that this is one of the most important chapters of the biology of cancer, to address the inflammatory status of the cancer cell environment.
My question is about implementation of anti-inflammatory drugs to reduce aging process, or to reduce speed of aging process. For example, some antibodies against interleukin 1, 6, some lymphocytokines.
So I do not hear very well. So. No, okay, but you can.
I can translate.
Yes. No.
Does not translate direct.
Yes.
Basically, I was asking, do you know anything about possible approaches to prolong life or reduce inflammation using anti-inflammatory drugs? So how they are prospective for?
This is another great point. Thanks for this question. The difficulty, well, I showed you a way that you can do relatively very easy. For example, change your diet, or not change. So, for example, following a Mediterranean diet. Mediterranean diet decrease inflammaging and rejuvenate the people. So this is for sure. Now, what I saw, there are recent papers, not mine, from other big groups that showed the same. So now there are at least 3 or 4 other papers that show that the Mediterranean diet rejuvenates. Okay, so why not? Oh, yes, no, this is yes. This is very interesting. I just asked a professor that I know in Bologna, not in Bologna, Reggio Emilia, to start something using monoclonal antibodies that are used in some diseases, anti-IL-6, anti-IL-1, and so on. They are nowadays used only for specific diseases. But according to Inflammaging, we could use these monoclonal antibodies against some of the most important inflammatory molecules to try to delay aging. This is a new idea. It would be great if you could follow this in Moscow, for example, and is what in science is very important. So something that a drug that has been used for a specific target to change the target. So this is very elegant. So something that is against some rheumatic diseases, for example, you can use for aging. And this is a very hot topic, but of course you must be very careful because a major message of my talk is that inflammation is good. So centenarians are centenarians because they are inflamed. Not much, but they are inflamed. So it's very difficult to know how much you have to decrease inflammaging, for example, with this approach, this new, very powerful approach using, for example, monoclonal antibodies against inflammatory markers, inflammatory compounds. So it's something that has to be studied the next year. Very good question. You are very well prepared here in this fact, according to the quality of the question. We will see if your question is good or not. Sì. Le do perfettamente ragione, ma di fronte a modelli iniziabili я подтвержу. Я президент Ассоциации итальянской ученых, которые остались работать в этой стране, в этом непростом. Sono il presidente dell'Associazione di scienziati italiani che sono rimasti a lavorare qui. Я сам жил в период, на который эта история чуть-чуть осложняла отношения в области науки между нашими странами. Ho vissuto personalmente il momento in cui questa nuova situazione ha reso difficili i rapporti accademici и scientifici tra Italia и Russia. Я знаю, и вы знаете, что как строже это негативно смотрит наша страна, эта страна. Lei sa come me che in questo senso è molto più aggressivo negativamente l'atteggiamento italiano per quello russo. Но все равно наука нельзя ни функционировать, ни создать науки и экономического сотрудничества фальшивые барьеры. Наука живет. Sì, la scienza non ha barriere. Science has no barrier. Yes. Per questo io le chiedo, e questo in italiano, se vuole far parte dell'associazione, visto che lei è una rarità di uno scienziato russo qui, поэтому я прошу принять участие в тартissimo. Con molto piacere, with great pleasure. Chiedo di aiutarmi a fare il lobbying del Tkachuk, quello di andare, sono stato l'addetto scientifico 2 anni, quello di andare nelle varie università e dire perché sono stati interrotti i rapporti quando appunto scienza e accademia dovrebbero essere sopra le parti. Cioè. Я очень рад, что вы президент. Как я вам сказал 1,5 года назад, я буду очень гордиться снова начинать читать лекции, как профессор нейрохирургии у вас. У меня работы не хватает, я главный научный сотрудник. Добро пожаловать. Тогда это слово всех. Контракт у меня здесь. Sì, sì. La sua presenza mi ha aiutato molto. No, I would like to answer to your question. No. I would like to give few words. So I started the mega grant when the relationship were quite good. Sorry. Yeah. No. But then the COVID, there was a COVID, and it could be impossible for me to come here. And now this situation of the Ukraine war. I am very clear. In all my life, I showed you that I have collaborated with Americans, with Chinese, with Russian people. So for me, no border. So this is what I think, and I hope that in the next future really, everything will, the peace will arrive. But the message for the young people is that I definitely think that science is over political, over borders, and has that the political situation should have no effect on our collaboration and so on. I was, for example, in the States. I can tell you a story. Sorry, I was in the States when there was they asked me to come to the National Institute on Aging to start a new project with them. And after a discussion, we went to the restaurant with the people of the NIH who invited me. And it was a period where there was the recounting of the vote between the two, Gore and Bush. Okay, so I was, there were all friends. And so talking, I said, well, there was a guy said, well, maybe it could be interesting to have a neutral observer in your election in the States, because there are these big problems. And to me, I said, well, for example, why not to ask some Russian people to come here and check? There was immediately the atmosphere changed immediately, and the person who was inviting me said, Claudio, sorry, but what you said, don't do it another time, because it will be difficult for me to invite you again. No, this is just to give you an idea that all the people are sensitive for what happened politically. And my star is that I am over this type of problem. Not because I don't know the problems. I know the problems, but I from what I can do, I am here, and I think that this is the best example that I can do.
Let me add something. This is a very good new atmosphere. Trying to understand that when the association was formed, there were Italian backing, and Russia were doubtful. Now we have the opposite. And as a person like me, and also my colleagues, to live here, have just one passport, and love Russia, is a difficult target. Меня учили любить эту страну, когда мне были 10 лет. И уже 10 лет давно я был юнионером. Поэтому менять не могу. Иногда бывает то, что, ну, это хорошо говорила какая-то девушка, которая сказала, хорошо, но чуть-чуть не надо. В России всегда есть много спряченных друзей, таких друзей мало. Их надо ласковать. И спасибо вам за ваши слова, профессор Ткачук, я вам доверял. Professor Francheschi, once again I'll ask you to be part of our team. We have people that won mega grants. There was a nice colleague, I'm from Tor Vergata, yes. And there was a nice colleague who won a mega grant. Now he disappeared after the SVO, the special operation. Yes. So I'll ask you to take part. We will know, and I will give you as a pleasure the place as a president. That would be nice.
Thank you.
этот вопрос. Во всем мире, к сожалению, так получается, может быть, к счастью, другой половина населения, мужчины живут меньше женщин. И мы понимаем, можно приводить очень много различий, и хромосомный набор, и X-хромосомы, которые, так сказать, женщину обеспечивают в большей степени от заболеваний, различных других. Но так получается, что женщина живет дольше, но последние годы ее связаны с тем, что резко снижается ее качество жизни. Мужчины живут меньше, чем женщины, но качество жизни не так резко снижается. Все-таки есть ли возможность, как считает профессор, что вот это неравенство, да, условно говоря, гендерное, на каком-то этапе можно было бы, так сказать, привести, так сказать, в равенство. Я за мужчин. Я хочу больше любить женщин. А так мы умираем, они останутся. Они без нас остаются.
Significant disparity in average life expectancy between men and women, and we know we can speculate that it's because women get double X chromosome that prevent inflammation, that women have more stable genome. But the fact is that women, even living longer than men,
Yeah.
they suffer
all over the planet.
Yeah, all over the planet, yes, but they suffer from drastically decreased level of life
After menopause. After.
At their late life. Men, on the other hand, got shorter lives, but they don't suffer
In better shape, yes.
Yes, severe. And Professor Malov asks, what do you think? How we can eliminate this gender inequality?
Well, probably. Well, I wrote some paper and speculation about this, which is very important. In particular, I published a paper with an Italian demographer. We looked at the historical data in Italy and in particular in Sardinia. So about 2 centuries ago, women in Italy and also in Sardinia usually had a lot of kids, about between 7 and 9. So this, and the average life expectancy of women was a bit lower than that of men. Then, during these 2 centuries, the number of children per women started to decrease, and accordingly, the life expectancy of women increased, so that there is a clear inverse correlation between the number of children and the life expectancy of women. The best is to have between 2 and 3 children. But also women who have no children, these are studied in nuns, also women without any child do have a small advantage. So the increase of life expectancy in women is inversely related to the decrease in the number of children. These are powerful historical data, because in Italy we have a lot of data set, and so they are very, very powerful. So this would suggest that the life expectancy is deeply related to the fact that women give birth to young babies. Why women with 3 children live longer than men? This is another. There is one possibility. Is that one? women usually do have more diseases in old age, more diseases than men. And they live longer. Why? Because probably one possibility, theoretical, but probably possibility is that there is an adaptation of their body to a low level of defects. So the defects are not simply things that accumulate, but if you have a defect, biochemical defect, the body reacts and try to re-establish the equilibrium. So the idea is that this is related to the so-called hormesis. Hormesis suggests that if you give a person a poison, small amount of a poison, eventually you become resistant to a high dose of the same poison. Okay? Mithridate, remember. So the same can occur in the body of women. So small amounts, a number of small defects can increase the capability of the body to react. So this is an hormetic effect, and is a powerful hypothesis that maybe you can follow. It's not so well known, but this can be an answer to your this is very important question. So theoretically is extremely important. Thanks for the question.
I think we have time for one last question.
Thank you, Professor Franceschi, for the very informative and interesting lecture as usual. So my question is that you demonstrated very clearly that the age-related biomarkers, sometimes heterogeneous at advanced age, doesn't mean that the mechanism of adaptation of aged people are also very heterogeneous and it is impossible to formulate it. Well, aging theory, biology of aging, because mechanisms are really very heterogeneous. And the related question, which is more practical, if we study the different cohorts of patients, it seems that the centenarians are more adapted to the stressors.
Yes.
And what about the pre-centenarians patients? How to distinguish the people who has benefits from the adaptation mechanisms from these patients who has some damage from the same stressors, and how to compare them in studies?
This is a very important question also, and we discussed just before coming here, Misha, before coming here, we discussed with Gurban exactly this point. So, how to measure resilience? So, and the idea is the following. Suppose that you have a ball which is here. In order to take the ball out of this place, you need some energy because the ball has to go out. So this is the attractor. This is your attractor. So we could try to measure how. So giving a stress to a person, we are thinking to measure how much a person, how much time it takes to revert to the same. So if I give a push to a person, the person say make like this and then arrive to the same position. In all the people like me, we can fall down. Okay, so this is the same. So this can be measured. So the resilience and so how can you remain in your attractor? So your place. So this could be another very important measure of resilience, which could predict your adaptability. Because the point, as you raised, is can we measure the adaptability of a person supposing that, for example, centenarians are the people who adapt better to stress. So this is a point. But there are mathematicians like Misha or Gorban, and it's up to them to do the job. Maybe no, in the future, and in a couple of years, maybe we can come here and tell you something about this point. Very important, very important. Would be very interesting to measure, to have this type of measurement of resilience, being inspired by this mathematical approach to the attractors.
Может, вы когда подойдете, поможете с переводом. Я не хочу по-английски плохо говорить. Мне не пристало на своем факультете демонстрировать свое произношение не английское, поэтому, если можно, да? Дорогие коллеги, мы прослушали очень полезную и, в общем-то, глубокую лекцию. Если будет время, будет возможность посмотреть на эти материалы, получив оттиски или слайды, я бы очень рекомендовал и это сделать, потому что здесь очень многие аспекты, важнейшие продления жизни человека были затронуты в результате очень большого многолетнего труда, и много мыслей рождают. Если позволите, поблагодарю лектора и могу сказать, мне было очень тепло, что узнать, что ваша альма-матер – это Болонский университет. Это первый в Европе университет, с которого началось образование в России, в Европе, во всем мире. Простите, в Европе, конечно. 800 лет назад. И мы это помним, мы знаем. В будущем году мы отмечаем 270 лет Московскому университету. Вы видите, насколько мы моложе. Спасибо за комплименты. Но мы продолжаем это образование. Мы идем из Болоньи, помним и уважаем и шлем приветствие вашей альма-матер. Я хочу сказать, что этот материал богатейший, он, конечно, дает возможность поразмышлять и заняться серьезным исследованием. Если позволите, я расскажу, мне вспомнилось, как я в своем кабинете, он был тогда на первом этаже, солнышко пригрело, я открыл форточку, и приближалась весна. И вдруг прилетели ласточки, сели на провода, сотни, и стали чирикать друг дружкой, разговаривать. Я решил, весна, все, дождались. И в это время стучат в дверь, входит студентка, а у нее она в мини-юбке. До этого девочки не надевали мини-юбки. И я подумал, что вывод сделает поверхностный наблюдатель, что когда прилетают ласточки, девочки укорачивают юбки. А можно сказать, когда девочки укорачивают юбки, тогда и ласточки прилетают к нам. На самом деле это корреляция. И такое исследование в медицине, они часто корреляционные, сообщения очень важные, но дальше должно быть исследование. Что это? Биологические часы, встроенные и в ласточек, и в девочек, с точностью до 1 дня. Или солнышко пригрело, тепло стало. Или продолжительность дня увеличилась. И много других гипотез. Экспериментировать на девочках нельзя. А на ласточках можно. И мне кажется, что вот это материал богатейший для того, чтобы поставить задачу перед исследователями, которые хотят понять механизмы и потом воздействие на эти механизмы продлить жизнь человека. Вот. И поэтому это очень важно вот это поговорить с теми мудрецами, которые жизнь свою посвятили тому, чтобы накопить материал медицинский, который дает возможность потом как-то вот развиваться медицинской науке уже направленно, ставя нужные опыты в нужной области. Я еще раз услышал для себя очень важные наблюдения и о метилировании, причем хочу обратить, оно точнее отражает возраст, чем длина теломеров. Вы не возражаете? Нет? Мне так кажется, что я читал. А метилирование мы знаем, где и зачем. И есть препараты, их в онкологии уже применяют, которые вызывают деметилирование, которые вызывают метилирование. Есть препараты, действующие и на ацетилирование гистонов. И это мы можем проверять на животных моделях, конечно. А это приходит уже в медицину. Кроме того, речь шла о том, что есть тканеспецифичные маркеры возраста и обновление в этих тканях идет по-разному, в зависимости от генетики, от эпигенетики, в зависимости от пережитых болезней. И, в общем-то, это тоже предмет для изучения. Ну, а микроокружение – это третий фактор. А четвертых я не знаю, никто не знает, мне кажется. Генетика, эпигенетика, прожитая жизнь, перенесенные болезни. И окружение, микроокружение, о котором тоже наш лектор так хорошо рассказал. И вот эти три основные темы, вот они очень важны, конечно, и продолжительности индивидуальной, ну и в состоянии здоровья тоже очень важны. Мне хочется поблагодарить всех, кто организовал, придумал нам встретиться сегодня. И я вот еще раз напоминаю, что в национальном проекте, я уж скажу, под продление жизни и в одной строчке и регенеративную медицину выделено 11 миллиардов 9 миллиарда рублей в этом году. Ну, товарищи ученые, экспериментаторы, врачи, ну давайте на это обратим внимание и постараемся что-то сделать общими усилиями. В одиночку никто ничего не сделает. Ни один университет ничего не сделает. Когда-то в студенческие годы я прочитал у Энгельса, Фридриха Энгельса, мы тогда читали их, классиков. Он сказал, меня поразило и не понравилось это, я не поверил. Но сейчас я понимаю, как это мудро. Он сказал, что потребность общества ускоряет развитие науки в сотни раз быстрее, чем сотни новых университетов. Вот потребность есть. Раз общество объявило такой проект, есть потребность. Она к нам обращена. Еще раз спасибо вам за прекрасную лекцию. Профессору Сталлоне отвечу непременно. Он затронул, я не верю. Вы знаете, и наш лектор сказал, что наука открыта для всего мира. И искусство открыто для всего мира. Когда-то русские ездили учиться рисовать и ездят до сих пор петь, учиться в Италию. Когда-то итальянцы к нам приезжали кое-чему учиться. Например, вы стали прекрасным нейрохирургом, обучившись у нашего профессора Коновалова, да? А в начале 20 века это Мекка была для европейской медицины России, правда? Когда здесь была лучшая в мире медицина, и как наука, и как практика. Это будет продолжаться. Никаких заслонов никто не поставит, это естественно. И мы готовы и сотрудничать, и встречаться. Вот. И большое вам спасибо, что вы хотите преподавать. Надо найти клеточку, надо в программе найти нужное место. Я всегда готов. Запретов у нас нет ни для итальянцев, ни для кого-то. Вот. Спасибо, что вы по-прежнему сохраняете любовь, уважение к нашей стране. Спасибо.